So welcome to the second demonstration video where we're looking at virus particle image analysis. So in this video, we're going to go through standard steps for analysis that you could use with any tool. Here we're using R and particularly the Bioimage package, where we're going to pre-process some image, import them, adjust them, then we're going to segment them and correct them. We're going to do quantitative analysis on the number of virus particles that are present to look at stats related to them. And we're going to use R to do that. So we're going to use the Bioimage and Imager packages. We're going to look at the number, size, and areas of virus particles. So install that package if you haven't already. And now to achieve that, we're going to move over to R to engage with that more fully. So let's check that these two packages are installed and read in the image, which is in our file. So we could change this to any image that's of interest. We're just arbitrarily going to use that one initially. So what we could do maybe is try A3 in this example. And we're going to store that as the file image, and therefore we can use EVI, the display function, to view that image. So you can see there we just have a few virus particles. So maybe let's look at image one. It was nicer because I had well, let's try 3 then and stick with that. And then we can make a negative image of it as well by taking the maximum away from the values. And we can also look at the histogram of intensities. So you can see there, so there's obviously a huge amount that are very, very high. If we change that to 150, we can kind of see more better the range of values there. So really any threshold is fine for this analysis. So if we change that to 0.5, and then we can convert that into binary image and directly see the number of independent virus particles. And remember, this is later on in the experiment, so there's fewer virus particles than if we did the first image A1. So this should be at 24 hours. And therefore we can identify the ones that are independent using the print function to identify using the BW label function, black and white labeling function, to identify the number of particles, independent virus particles that have not merged that are present. So we get 10. If we went back to step 2, again, we look at the histogram, which is key. Again, 0.5 is fine here. And we look at the number of virus particles present, there's 17. If we went back to part 1, again, we look at the histogram more carefully. That's more varied. So perhaps 0.4 would be better there. To estimate the number of objects, we get 35. I think you see, so if in the original, I had 38 because I used a more generous threshold. So this is why the thresholding has a strong effect on what we get at the end. So we have an estimate of 35 based on this analysis. What we can do then is assess the features based on our image there, and measure their size and area. So if we look at this data frame, we can get the area, the perimeter, the radius mean, and other metrics. We're going to focus on the first two. So if we get a summary of them, what we can see down there is that the area, the median area is 7, max is 40, minimum is 1, and that's for the image on the right. Look at the variance there. We got a variance about 10. It was higher, but we have more virus factors and less stringent, so that all these things kind of correlate. Now we do it for the cell. So this is looking at image 2. So if we look at that one, we can see the cell more clearly. If we change that to image A3 for the cells, the different channel, it is changed a bit. So here we're expecting we have 2 cells with this background flux from the virus particles. So a threshold there of maybe 0.3, perhaps. And if we do that again to estimate the number of virus, we got 31. So this is not very good. We need to actually blur that or use a more stronger threshold. to get something that's more effective. So we change this to a very high value of 0.8. We see that that doesn't work either because all the cells just kind of merge into one. So that's not really going to work. So what you ideally do here is blur that image. So you see there, we get a 0.5, 0.45, we get a value of 7, which is closer to our true approximation of 2. So if we look at the object props 2 containing the stats for these ones, you can see 2 large ones, and the rest are just virus factories. So we can get the information about them. So we have a mean of 429, a standard deviation varies a lot. So what you can try then is reapplying this to the code. And what we'll do in the lab then is use the gblur function to look at how we can optimize our capture of the cells in particular using blurring. Okay, so that's the end of this video.